Welcome to the deep dive. When you think about the medicines we take, how do we actually know, like really know, that every single pill or dose is going to work properly and be safe?
Yeah, it's a huge question. And the answer is, well, it's complex. It goes far beyond just checking the final product off the line.
Right, it's not just pass-fail at the very end. We're looking into something called quality by design today, QbD. It sounds like a smarter way to build quality in from the start.
That's a great way to put it. Instead of just hoping the cake turns out right and maybe testing a slice, QBD is like deeply understanding the recipe, the oven, the ingredients, everything, so you're confident before you even bake it.
Okay.
So our mission today is to unpack how this QBD approach actually gets used. We'll be focusing quite a bit on a tool called Design of Experiments, or DOE.
DOE, got it.
And while our sources don't spell out the FDA's exact stance on QBD and specific applications, we can certainly talk about how the whole philosophy aligns with what regulators are looking for: safety, efficacy, consistency.
Makes sense. And we've gathered quite a range of materials for this: scientific papers, regulatory info, some case studies. Should give us a good picture.
Definitely.
So let's dive in. At its heart, what is QBD? What are the core ideas?
Okay, so fundamentally, QBD is a systematic approach to drug development. There's this key guideline, ICH-Q8, that defines it really well.
ICH-Q8.
Yeah, it's kind of a big deal in the pharma world. It calls QBD a systematic approach to development that begins with predefined objective and emphasizes product and process understanding and process control based on sound science and quality risk management.
Whoa, okay, that's a mouthful. So breaking that down,
Hmm.
it starts with knowing what you want to achieve.
Exactly. Predefined objectives.
Yeah.
And then the big emphasis is on understanding. Understanding the product, understanding the process used to make it, and controlling that process. It's all built on science and managing risks.
So it's less about following a fixed recipe blindly and more about knowing why the recipe works.
Precisely. You have to grasp the links between how the drug actually performs, its quality features, and the nitty-gritty of the manufacturing steps. It's not just, does it work?
Right.
But why does it work? And how do we keep it working reliably?
Okay, that leads us nicely into this design of experiments thing, DOE. That's the tool for gaining that understanding, right?
Spot on. DOE is absolutely central to QBD. Think of it as super efficient experimentation. Instead of the old way, changing one thing, seeing what happens, changing another.
One factor at a time, yeah.
Slow, very slow, and you miss interactions. DOE lets you systematically vary multiple inputs process parameters like temperature or mixing speed, maybe attributes of the raw materials all at the same time in a structured way.
Okay. And the goal is?
The goal is to map out how these changes affect the really important characteristics of the drug, the critical quality attributes, or CQAs.
CQAs, so that's like its purity, how strong the dose is, how quickly it dissolves, stuff like that.
You've got it. Purity, potency, stability, dissolution rate, all the key things that define quality and performance. And we actually have a great example from our sources involving a drug called Torcetrapib.
Torcetrapib. Okay, what do they do?
They used DOE pretty extensively during its development. These experiments were crucial for identifying the CQAs for both the active pharmaceutical ingredient, the API.
The main drug substance.
Right. And the final drug product, like the tablet. The DOE helped them understand the relationship between the process steps and those critical quality aspects.
So what kind of things did they figure out using DOE for Torcetrapib?
Well, one key outcome was establishing the design space. That's a term from the ICH guidelines again.
Design space sounds like the operating window.
Exactly. It's the range of process parameters and material attributes where you've proven that quality is assured. It's not just one single setting, but a defined multidimensional space. For Torcetrapib, DOE helped them see how different factors affected impurity levels.
Impurities are always a big concern, I imagine.
Huge. So they ran experiments comparing, for instance, using sodium carbonate versus sodium phosphate to control a specific impurity during a reaction step.
And did one work better? I heard there was a bit of a surprise there.
There was. Initially, sodium carbonate looked better for controlling that impurity, but the experiments revealed something unexpected. If they let the reaction run too long with the sodium carbonate, or maybe use too much, the whole reaction could just stop working.
Really?
Why? It turned out there was a secondary reaction happening. When the ratio of reactants hit a certain point because of this side reaction, it basically used up one of the essential starting materials too quickly.
Wow. So the DOE didn't just confirm things, it uncovered hidden complexities.
Absolutely, that's the power of it. Now, interestingly, this stalling didn't actually ruin the batch in their case. They had a safety net built in. They had what's called a process acceptance range, or PAR. Basically, they knew they could remove any leftover starting material in a later purification step, but finding that stalling phenomenon, that gave them much deeper process understanding.
I bet. You wouldn't find that just by testing the end product.
No way. The DOE results also let them rank which variables had the biggest impact on getting rid of impurities. And when they looked at the crystallization step later on, DOE helped define the precise boundaries for that part of the design space, too.
So really mapping out the safe and effective zone for manufacturing.
That's it. And this enhanced understanding you get from QbD, powered by DOE, it directly leads to better control over those critical quality attributes.
Like keeping those impurities consistently low in the Torcetrapib example.
Exactly. Optimizing the process based on that DOE data meant they could confidently keep impurities within safe, acceptable limits, batch after batch.
And does this apply to other CQAs too, like how well the drug dissolves?
Yeah. Same principle, absolutely. While the sources don't detail a specific Torcetrapib DOE for dissolution, understanding how formulation factors like particle size or binders in a tablet and process parameters affect drug release is a classic QbD DOE application. You control those inputs to ensure the drug dissolves predictably.
Sometimes the dissolution rate itself is a specification, isn't it?
It often is. It can be a key quality standard directly linked to how the drug will be absorbed in the body.
It really emphasizes that QBD isn't just about observing correlations if we do X, Y happens. It's about the why.
Precisely. That unexpected reaction stalling with Torcetrapib is a perfect example. It forces a deeper dive into the underlying chemistry and physics of the process.
So how does all this fit with the regulators?
Yeah.
Like the FDA, you mentioned our sources are a bit light on specifics here.
They are on direct quotes about QbD and NDAs, but we can definitely infer the alignment. The FDA's core mission, above all, is ensuring drugs are safe and effective for patients.
Right. That's job number one.
And the principles of QbD, understanding your product, understanding your process, controlling your process based on science and risk, they feed directly into that mission. It's exactly the kind of proactive approach regulators want to see.
So when a company submits a new drug application, an NDA, the FDA looks closely at the manufacturing part.
Oh, absolutely. They scrutinize the manufacturing process description, the controls in place. They conduct pre-approval inspections, PAIs, on-site pre-approval inspections. They go to the manufacturing facility to verify the information in the application is accurate and, crucially, that the site is complying with current good manufacturing practices, CGMPs.
CGMPs, right?
Yeah.
Baseline rules for quality manufacturing.
Exactly. And even earlier, during the investigational new drug phase, the IND phase.
Before human trials start.
Correct. Even then, regulators focus on ensuring the manufacturing process for the trial drug is reproducible and that the drug is well characterized. They pay close attention to impurities and potential contaminants right from the start. QbD provides a framework to generate that characterization data systematically.
So QbD helps build the evidence package that gives the FDA confidence.
That's a good way to think about it. It demonstrates a deep level of understanding and control, which inherently reduces risk. It aligns perfectly with the goals of CGMP. You can almost see QbD as the scientific engine that helps you design and implement truly robust GMPs.
It helps you figure out which parts of the GMPs are most critical for your specific product and process.
Exactly. It tells you where to focus your control strategy for maximum impact on quality.
Okay, this has been really insightful. Let's try and sum up the key takeaways from this deep dive on QbD.
Sure. I think the main point is that QbD represents a shift towards a proactive, science-driven way of ensuring pharmaceutical quality. It's not an afterthought. It's built in.
Using tools like design of experiments to really dig deep into how manufacturing processes work.
Right. To gain that fundamental process understanding. And that understanding is what allows for effective control over those critical quality attributes, the things that really matter for the patient.
And while we didn't have specific FDA mandates in our sources today, the whole QDD philosophy seems perfectly in tune with regulatory expectations for safe, effective, and consistently produced medicines.
Definitely. It's about building quality, safety, and efficacy from the very beginning, based on solid science and risk assessment. It's a framework for developing a robust, reliable manufacturing process.
Which ultimately means better, more dependable medications for all of us.
Yeah.
Makes you appreciate the science that goes into ensuring quality.
It really does, and it leads to a final thought, perhaps. As pharmaceutical manufacturing gets ever more complex, Think biologics, cell therapies.
Yeah, cutting-edge stuff.
How will QBD itself need to evolve? How can the systematic approach adapt to meet the challenges of these new modalities and technologies? That's something worth pondering.
A fascinating question for the future. For listeners wanting to dig even deeper, checking out those ICH guidelines, particularly Q8, and looking for more QBD case studies would be a great next step.
Absolutely. There's a wealth of information out there once you start looking.